Welcome to Academic Express. In the biological taxonomy of the animal kingdom, birds belong to the phylum Chordata and the class Aves. Ornithology is the general scientific study of these species, but poultry science is a much narrower discipline with a very rigid boundary. This photograph shows cormorants used for fishing. While they serve a human purpose, utilitarian birds like these, along with captive songbirds and sporting falcons, do not meet the definition of poultry. True poultry refers to species kept and genetically managed exclusively for human benefit, specifically for the harvest of meat, eggs, feathers, and down. Poultry science applies selective breeding to convert avian biology into a specialized agricultural technology. Domestication is a sustained, multi-generational relationship where humans assume control over an animal's reproduction to secure a predictable resource. Early human societies achieved this by exploiting natural bird behaviors, whereas modern scientists actively rewrite their cellular biology. Thousands of years of intervention have modified avian genomes to create highly divergent, continuous production biological systems. The modern chicken is the result of directed evolution, a biological system modified specifically for human requirement. This diagram illustrates the timeline of avian domestication, beginning in the Neolithic period. The earliest branches trace the domesticated chicken. Archaeological evidence is contested, placing its initial domestication from the red junglefowl between 3000 and 10,000 BCE. Later branches show the independent domestication of waterfowl, including mallard and Muscovy ducks, along with European greylag and Chinese swan geese. Other branches detail outlier species, pigeons kept for homing, guinea fowl in Africa, Japanese quail, and turkeys native to the Americas. The most recent addition is the ostrich, domesticated via a hybrid cross in late 19th century South Africa. This table catalogs these disparate domestication events. The putative time of domestication column shows the precise chronological spread across these species. Across these disparate taxa, humans applied selective pressures to restrict breeding, eventually forcing compliance and maximizing agricultural yield. This photograph shows the male red junglefowl, the wild phenotype of the domestic chicken. It inhabited the dense forests of Southeast Asia. Bamboo follows a specific masting cycle, blooming and dropping massive amounts of seed simultaneously, roughly once every 50 years. The red junglefowl evolved a hyper-reproductive response to this calorie surplus. Early humans essentially hacked this 50-year timer. By providing the birds with a constant food supply, they simulated a perpetual bamboo bloom. Constant caloric intake tricked the bird's neuroendocrine system into entering a state of continuous, year-round ovulation. Scaling this required a shift in how eggs were hatched. Ancient Egyptian engineers designed massive mud ovens that functioned as artificial incubators, capable of holding up to 4,500 eggs at once. Mass artificial incubation severed the biological requirement for maternal care. Selective breeding could then remove the neuroendocrine trait of broodiness, as the hens no longer needed to sit on their eggs. Manipulating the bamboo masting response and outsourcing incubation were the first steps in converting a wild animal into an industrial technology. While chickens share a monophyletic origin in the red junglefowl, their modern genetic makeup is a mosaic, patched with traits borrowed from other species. This diagram illustrates cross-species introgression. For example, the yellow skin pigmentation on the legs of modern chickens was spliced into the genome through interbreeding with the gray junglefowl. Another major shift involves the thyrotropin receptor, or TSHR. A specific missense variant in this gene alters hypothalamic expression, which is required to regulate persistent egg production. Archaeological DNA proves this TSHR mutation only became fixed in the poultry population within the last 500 years, rather than 8,000 years ago as once assumed. We also see shifts like the ectopic expression of the CYP19A1 gene. This causes aromatase production in the skin, resulting in males' anthropogenic genetic shifts. Catalog genetic engineering program, rewriting the metabolic blueprints of these birds. This photograph shows the transition to the era of scientific selection, where village farming was replaced by scaled, uniform production in industrial poultry houses. This intensive selection caused extreme physical divergence. The tiny Sarama chicken weighs under 250 grams, while the Jersey giant can reach nearly 7 kilograms, demonstrating the elasticity of the avian genome. In 1945, corporate breeding programs pushed broiler weights from 1,400 grams to over 2,400 grams, specifically prioritizing rapid growth rates. This comparative image provides visual evidence of this morphological divergence. The slender, colorful junglefowl at the top stands in contrast to the heavy, white Plymouth Rock at the bottom. Breeders masked complex plumage colors by adopting the recessive white genotype. White feathers lack dark pin feathers, making post-slaughter processing cleaner for the consumer market. 20th century selection prioritized rapid muscle accretion and processing efficiency, largely disregarding natural survival mechanics. This photograph of a bourbon red tom establishes the baseline anatomy of the domesticated turkey, a species native to North America. Unlike chickens and ducks, turkeys feature bare head and neck appendages. These anatomical structures evolved to aid thermoregulation in hot, dry climates. Modern selection for massive breast meat yield has created extreme sexual dimorphism. In commercial populations, males can grow to 18 kilograms, while females reach only 9. The mechanical consequence of this dimorphism is severe. The top heavyweight distribution of commercial turkeys makes natural mating physically impossible. Because they cannot reproduce naturally, the continuation of the commercial turkey species relies entirely on human-administered artificial insemination. Scientific selection pushed avian physiology past its natural boundaries, to the point where the animal can no longer perpetuate its own species without human intervention. This schematic illustrates the biological tax of modern hyperproduction. The cloaca functions as a shared funnel for reproductive output and waste. Forcing this system into daily overdrive risks a trapped egg, which blocks the excretory pathway, causing lethal constipation.